My name is Patrick Muldown. I'm a PhD candidate in the lab of Niall Rawlinson at the University of Toronto, and I was very fortunate in, in 2020 to receive a scholarship to attend the World Congress of Herpetology in Dunedin, New Zealand, and present my natural history-based research on salamanders as rich prey for carnivorous plants in a nutrient-poor bog ecosystem. Salamanders are well known for contributing a disproportionate amount of biomass in the forests of eastern North America. This illustration here just shows an example spotted salamander in relation to other typical forest wildlife, and as you can see, its scaling is absolutely enormous. And that really speaks to the ecosystem roles that these animals play, certainly dwarfed by something like the invertebrates, which of course form the base of the food chain, but nonetheless still kind of punching above their proverbial weight here in their ecosystem services. And through the aquatic to terrestrial life cycle of these salamanders, they contribute in a lot of ways, and perhaps most pertinent of which is presented here has to do with trophic regulation. That is, the contribution these salamanders make both as predators and as prey. The research that I presented in Dunedin was a bit of a curious example of how salamanders form prey, a very much unexpected manner for that matter. The spotted salamander, as seen here, is widespread throughout eastern North America, and that yellow star on the range map shows where I study them towards the northern end of their range limit in an area called Algonquin Provincial Park. We have a long-term mark recapture study with these salamanders, and we're intensively working on some of their demography. But in the process of doing that, we stumbled upon something that was very surprising. And that surprise involved these plants. This here is called the northern pitcher plant, and seen once again with the range map there marking our study location in south central Ontario, province of Canada, is where both the spotted salamander and these carnivorous plants overlap. These plants grow in very nutrient-poor bog environments, and they're uniquely adapted to acquire nutrients through a secondary source. So rather than simply drawing nutrients from their roots, as you typically think of a plant, these carnivorous plants trap and catch a huge abundance of invertebrate life. However, although these plants have been incredibly well studied over the past century, vertebrate prey has been recorded extremely infrequently. And thus the surprise when we began to find salamanders turning up as prey in these plants. We set forth a number of questions, but really this is, to date, given that it's such a recent discovery that we turned up, we don't have a lot of concrete data, and really it's a backbone sort of natural history study, but we certainly do have a number of questions. I think questions that we're charting the course forward with as we go. So, what we observed and what I presented in Dunedin is that 20% of our surveyed plants, of which there were 90 plants in our survey population, had caught metamorphic salamanders that were outbound of the breeding pond, and they're getting trapped and they die in these plants, which can then subsequently absorb their nutrients. So one question we have, for example, are there habitat or plant-specific characteristics that predict the salamander, the capture of salamander prey? There's some obvious hypotheses that sort of jump out at this. One is that maybe the salamanders are drawn to plants because they too are seeking food, mostly in the form of insects. An alternative hypothesis, sort of a null in a way, is maybe the plants just so happen to catch these salamanders on their trajectory out of the ponds. There are many other hypotheses that we're under investigation and formulation, for that matter, right now. Another question we have, is sort of grim as it might sound, is following entrapment, what is the cause of death of these salamanders? We've seen some perish in as little as 24 hours, and others that have lived over a month within this soupy fluid mixture that's contained in the plant. And once again, we don't have a definitive answer here, but rather just lots of different pathways for hypothesis-driven work going forward. So it may have something to do with the physiology, upsetting the physiology of these amphibians and sensitivities of their skin or upset of their homeostasis, for example. These plants are noted as releasing enzymatic cocktails into their fluids. And as such, maybe there's something related to that causes the general loss of orientation followed by death that we see in the salamanders and perhaps accelerates their decomposition. But anything from that to suppressed immune systems that we know about and recently metamorphosed salamanders may further compromise them once they're trapped by the plant. And we're also actively collecting temperature data to see if, in fact, these salamanders might get cooked, for lack of a better word, from exposure to extreme temperatures while trapped in these plants. Of course, too, there's a huge benefit to be had by these plants catching such a monumental nutrient source when most of the time they're catching small insects ranging from beetles to ants to small moths and butterflies. These salamanders contribute proportionately a lot to the nutritional balance of these plants. And so just to give a brief breakdown here, for every spotted salamander captured, which is represented as the black salamander figure with the yellow spots, it's equivalent in nutrition to one juvenile eastern or red-spotted newt, another North American species that's been recorded from the pitcher plants, or about a nutrient equivalent in nitrogen to about 125 ants or 500 small flies, Drosophila fruit flies just being an example here. As you can see, for every salamander that these plants capture, they gain a huge nutrient load. And so, what's kind of spawned as a study based in herpetology with these salamanders has begun to slowly convert over to a botanical investigation about the nutrient allocations and the benefits and ultimately the trophic interactions between these plants and salamanders. And again, I'll just mention, as we've uncovered so far, 20% of these plants were catching salamanders. But with increasing survey effort, we anticipate that number will probably go up. Just in the case right now with preliminary data collection, there's a lot more to come. That's quite exciting for what has really become a side project that's absolutely captured me. Just to keep this relatively brief, I think what's rather fascinating is that as a salamander, this animal's life is very uniquely tied to plants, right from beginning to end. You may know about the spotted salamander that it has a symbiotic or mutually beneficial relationship with a special algae that grows in their eggs. And a photo of that's represented on this slide here. That greenish hue in the egg capsule is algae, and that helps take in the carbon dioxide produced by the embryos and converts it to nitrogen to support development. It also breaks down toxic wastes, thereby supporting the salamander. So whereas this algae greatly benefits the salamander during its early developmental period, here we have, by contrast, another plant, in this case, a carnivorous plant that can take life away rather than be life-giving. So with that, I'll just wrap up this brief synopsis of the presentation here, and I'll thank you for listening. The scholarship to attend the World Congress of Herpetology in Dunedin in 2020 was wonderful. Of course, without that support, I would not have been able to attend. And by attending, not only was I very excited to share my work, but network with colleagues, meet other young scientists, and of course, meet those whose work that I really admire and respect. The conference, which I was able to attend because of the scholarship, it exceeded expectations by far. I mean, when I think of things like the inspiring plenary talks, the wonderful break sessions, which afforded this opportunity to talk with all those people I've been meaning to catch up with or meet and introduce myself to for the first time, all the way through to the banquet, which was absolutely terrific. The university in Dunedin was just such a wonderful venue for the meeting, and I heard so many people talk about how beautiful it was. Beyond that, things like the field trips and the opportunities to, of course, see the unique New Zealand herpetofauna was outstanding. And on account of this scholarship and, of course, some leniency from my supervisor as well, I arrived in New Zealand early and I stayed quite late. And that was fabulous, because not only did it mean through this scholarship I was able to attend the meeting directly, but I'd go on to help volunteer on an island conservation project in New Zealand, something that I'm particularly passionate about and where I would see my career going in the future, being involved with applied conservation initiatives with threatened or ultimately imperiled herpetofauna. So really it was additive. It was not just simply that I attended the conference and had these networking opportunities with professionals that would help advance my career. But not only that, I was able to gain experience as part of the process by joining on with field teams and enjoying the lovely country that is New Zealand in the process. All right, what's next? Well, I still do have a little bit of time left on my PhD studies, but I don't think I'll be leaving research anytime soon. I'm not quite certain which capacity that might take, whether it be academia or hopefully a research scientist position. With much of the networking that I was able to accomplish at the World Congress, I've maintained contacts since, involved in different international conservation projects, especially those on islands. And so that's where I'd love to see myself into the future, in a research scientist and applied conservation capacity. I'm very passionate about education as well, and sharing information about herps, especially given that they're a largely misunderstood and largely underappreciated group of animals. So if I could strike a balance somewhere between research and education, I would be very, very happy. And work with salamanders and turtles has been my focus to date, has been wonderful, but I see that branching out well into the future as well.